Welcome! Today, we're exploring about genetic mutations, what they are, why they happen, and their impact on living things.
Good morning, everyone! Today, we are going to talk about the introduction to genetic mutations. Introduction. Genetic mutations, alterations in the DNA sequence of an organism, are caused by errors in the DNA replication or exposure to harmful substances. These changes are fundamental to evolution, providing the genetic diversity that naturally selection operates on. Understanding genetic mutations is crucial for studying the genetic basis of diseases and developing new treatments. These alterations can vary from single nucleotide changes to deletions or duplications of entire genes. Mutations can occur spontaneously or be induced by exposure to mutagens, and their effects on an organism range from neutral to harmful or beneficial, driving the process of evolution. Genetic mutations are very helpful in many ways, including the evolution, genetics, and diseases. So, in evolution, mutations create new genetic variations which can lead to new traits and adaptations. These adaptations can help organisms survive and reproduce in changing environments, driving the process of evolution. On another hand, genetics, the study of genetic mutations, can help us understand how genes function and interact with each other. By studying mutations, scientists can identify genes involved in specific traits or diseases. And lastly, on diseases, many diseases are caused by genetic mutations. These mutations can alter the structure or function of proteins, leading to abnormal cellular processes and disease. Understanding the genetic basis of diseases can help in developing new treatments and therapies. The importance of understanding genetic mutations in biology, medicine, and evolution. In genetic mutations in biology, mutations are crucial in biology as they introduce genetic variation, which drives evolution and adaptations in populations. They can also lead to new traits and diseases, influencing an organism's survival and reproduction. The importance of knowing the genetic mutations in medicine. So, mutations are crucial in medicine because they can lead to genetic diseases but also offer insights into how these diseases occur, enabling the development of targeted treatments. They also play a role in understanding drug resistance, such as in cancer or infectious diseases, helping to create a more effective therapies. And lastly, we have the importance of genetic mutations in evolution. So, mutations provide the genetic variation that natural selection can act upon, driving the new process of evolution. By creating new genetic combinations, mutations allow organisms to adapt to changing environments and develop new traits.
Moving forward, let's delve into the different types of genetic mutations. What is a point mutation? A point mutation is a genetic mutation where a single nucleotide base is changed, inserted, or deleted from a DNA or RNA sequence of an organism's genome. Point mutations have a variety of effects on the downstream protein product, consequences that are moderately predictable based upon the specifics of the mutation. What are the types of mutation? Now let's talk about missense mutation. What is a missense mutation? It is a base substitution in which one nucleotide is replaced by another one that changes the codon in a way that a different amino acid is synthesized. Nonsense mutation. What is nonsense mutation? Nonsense mutation changes a codon that specifies an amino acid into a stop codon. Silent mutation. What is silent mutation? Silent mutation changes a codon that specifies a certain amino acids into a different codon that, however, still specifies the amino acid. Now shifting gears, let's explore insertion and deletion. What is an insertion? An insertion is the addition of one or more nucleotide base pairs into a DNA sequence. These can often happen in microsatellite regions due to the DNA polymerase slipping. What is deletion? Deletion is a type of mutation that involves the loss of one or more nucleotides from a segment of DNA. A deletion can involve in the loss of any number of the nucleotides from a single nucleotide to an entire piece of chromosome. This sequence is called frame shift mutations. A frame shift mutation refers to the insertion or deletion of nucleotide bases in numbers that are not multiples of 3. This is important because a cell reads a gene's code in a groups of 3 bases when making a protein. Frame shift mutations occur when 1 or 2 bases are inserted or deleted from the coding region of DNA. The result is a completely changed amino acid sequence and a very severe effect on the protein.
Variations and chromosomal mutation by OKWORI Judith Alice. So, copy number variations are alterations in the number of copies of a specific DNA segment within a genome. This variation can range from a few hundred base pairs to an entire chromosome, and they can be present in a single gene or multiple genes. CMVs are common type of genetic variations occurring in approximately 10% of human genome. Examples of copy number variation: deletion, duplication, and insertion. Example of deletion is, for example, Cru-de-Chart syndrome is caused by deletion on chromosome 5. Duplication, the example is Charcot-Marie-Tooth disease type 1A is caused by duplication on chromosome 7. Then for insertion, Williams-Boren syndrome is caused by deletion and insertion on chromosome 7. Impact of copy number variations. Copy number variation can have wide range of effects on individuals' health depending on the size and location of the variations. Some CMVs are associated with specific diseases while others have no noticeable effect. CMVs can also contribute to complex traits such as height, intelligence, susceptibility to certain diseases. Moving on to detection of CMVs. Detection of copy number variation can be done using the various techniques. 1. Chromosomal microarray analysis. This technique is used to scan the entire genome for CMVs. The next one is fluorescence in situ hybridization, also known as FISH for short. This technique uses fluorescent probes to detect specific DNA sequence on chromosomes. Then the last but not the least, next generation sequence. These techniques can identify CMVs by analyzing the number of reads for each DNA segment. Chromosomal mutations now. There are changes in the structure or number of chromosomes. This mutation can occur spontaneously or be induced by environmental factors such as radiation or certain chemicals. Types of chromosomal mutations: deletion, duplication, inversion, translocation. Deletion is a portion of chromosome is lost. Basically, this can result in the loss of one or more genes, leading to various genetic disorders. Duplication, a segment of the chromosome is copied and inserted in the same chromosome, resulting in extra copies of gene. This can lead to altered gene expression and potential developmental abnormalities. Inversion, a segment of a chromosome is flipped and reinserted, reversing the order of genes. This can disrupt gene regulation and potentially lead to developmental problems. Translocation, a segment of one chromosome breaks off and attached to the other chromosome. This can disrupt gene expression and lead to various genetic disorders, including cancers like chronic myeloid leukemia.
Now let's dive deeper into the causes of genetic mutations, which can be categorized into three main groups: spontaneous mutations, induced mutations, and biological factors. Spontaneous mutations happen naturally, without the influence of external factors. They primarily arise during DNA replication, which occurs every time a cell divides. During replication, DNA polymerase, the enzyme responsible for copying DNA, sometimes makes mistakes. These can lead to mismatches in base pairs or even small insertions or deletions of nucleotides. Although these errors are rare, they do occur randomly. When spontaneous mutations occur in reproductive cells, they can be passed on to the next generation, potentially influencing evolutionary changes. Next, we have induced mutations, which are caused by external environmental factors. One well-known cause is radiation. For example, exposure to ionizing radiation like X-rays or gamma rays can physically break DNA strands or cause chemical modifications in the DNA bases. Another major factor is chemical exposure. Substances found in cigarette smoke, certain industrial chemicals, or even some household products can bind to DNA, altering its structure and potentially leading to improper replication. In addition, ultraviolet or UV light from the sun can damage DNA by causing thymine bases to bind together, forming what's known as thymine dimers. These dimers prevent DNA from replicating correctly, which can lead to skin cancers and other issues. Finally, biological factors can also induce mutations. For instance, certain viruses insert their own genetic material into the host cell's DNA, disrupting normal gene function. One of the most interesting biological causes of mutations involves transposons, also known as jumping genes. These are DNA sequences that move to different positions within the genome. When transposons insert themselves into new locations, they may disrupt essential genes or regulatory elements, causing mutations. In some cases, bacterial infections can also create oxidative stress, leading to DNA damage. So, to sum up, genetic mutations can occur naturally through spontaneous errors, be triggered by environmental factors like radiation and chemicals, or result from biological agents such as viruses and transposons.
Mechanisms of mutations. DNA replication errors. DNA replication is crucial for cell division, where the DNA double helix unwinds and DNA polymerase synthesizes new strands by adding complementary nucleotides. This process ensures that each new cell has an identical copy of the DNA. However, despite proofreading mechanisms, mistakes can still occur. Mistakes can happen via mismatches. If DNA polymerase incorrectly pairs nucleotides, it can lead to point mutations. If not corrected, this changes the genetic code, potentially altering protein function. Insertions or deletions. These errors occur when the DNA polymerase adds or skips nucleotides, leading to frame shift mutation, which change the reading frame of genes, disrupting protein synthesis. Slippage. In areas which remove a short segment of damaged DNA strand, and DNA polymerase synthesizes a new, undamaged section. NER is crucial for protecting against skin cancers like xeroderma, fragmentism. Next, homologous recombination. When both DNA strands are broken, HR uses a sister chromatid as a template for accurate repair. This process ensures the restoration of the original sequence. HR is high-fidelity system crucial for maintaining genome integrity during cell division. Its dysfunction is linked to breast cancer. Lastly, non-homologous end joining. This system also repairs double-strand breaks but without a template. It directly joins the broken ends, making it faster but prone to errors like small insertions or deletions. NHEJ is vital for repairing breaks outside of cell division but can lead to mutations, contributing to genomic instability if frequently used.
Let's explore the effects of genetic mutations, which fall into three main categories. We have neutral, beneficial, and harmful. First, let's talk about neutral mutation. Neutral mutation, these are changes in the DNA that neither harm nor benefit the organism. As an example of a neutral mutation is a change in the non-coding regions of DNA, sometimes referred to as junk DNA. These regions don't code for proteins, so mutation here generally don't impact the organism's function. Although junk DNA was once considered useless, recent research suggests it may play a role in gene regulation. The mutation in these areas often remain silent. Next, we have beneficial mutation, which contribute to a positive changes in species over time. A famous example is lactose tolerance in humans. Originally, most humans could only digest lactose. as sugar found in milk during infancy. However, in some populations, a mutation allowed adults to continue digesting lactose, providing a valuable nutrient source. This mutation becomes common in societies where dairy farming was practiced. Another example is antibiotic resistance. Antibiotic resistance in bacteria, where a single mutation can allow bacteria to survive even in the presence of antibiotic, leading to more resilient strains. Finally, we have harmful mutations, which can lead to severe genetic disorders. For example, cystic fibrosis results from a mutation in the CFTR gene, causing thick, sticky mucus to build up in the lungs and digestive system, leading to chronic respiratory and digestive issues. Another well-known example is sickle cell disease, caused by a mutation in the HBB gene that distorts red blood cells into a sickle shape. This can cause blockage in blood vessels, leading to pain and organ damage. Interestingly, despite its harmful effects, sickle cell disease also provides a survival advantage against malaria, showing how even harmful mutations can have a complex evolutionary impact. So in summary, mutations vary widely in their effects. Some are neutral and go unnoticed. Some provide an advantage that can drive evolution, while others can lead to significant health challenges.
Today, we are going to talk about the genetic disorder caused by mutation, the single gene mutation, their examples, inherited patterns, and also the complex disorders. Genetic disorders arise when a mutation in a gene's DNA sequence interferes with its normal function. These mutations can be passed on from parents or occur spontaneously during an individual's development. So what is a single gene mutation? A single gene mutation is a change or alteration in the DNA sequence of one specific gene. This mutation can disrupt the normal function of the gene, leading to various genetic disorders, and can be inherited or occur spontaneously. Examples are cystic fibrosis. Cystic fibrosis results from a mutation in the CFTR gene, causing thick mucus to build up in the lungs and digestive system, leading to breathing and digestive issues. It is inherited in an autosomal recessive manner, meaning both parents must carry the gene mutation for a child to be affected. Cystic fibrosis follows an autosomal recessive inheritance pattern. This means both parents must carry a copy of the mutated gene. They are usually carriers without symptoms. If both parents are carriers, there is a 25% chance the child will have cystic fibrosis, inherits two mutated genes. 50% chance the child will have a carrier like the parents, or inherits one mutated gene. 25% chance the child will be unaffected and not a carrier. Males and females are equally affected, and the condition can skip generations if they are carriers but no affected individuals. Sickle cell anemia. Sickle cell anemia is caused by a mutation in the HBB gene, leading to abnormally shaped red blood cells that block blood flow, causing pain and anemia. It is inherited in an autosomal recessive pattern, meaning both parents must pass on the mutation for a child to have the disease. Both parents must carry one copy of the mutated gene, usually without symptoms. If both parents are carriers, each child has a 25% chance of having sickle cell anemia, inherits two mutated genes, a 50% chance of being a carrier, or inherits one mutated gene like the parents, a 25% chance of being unaffected and not a carrier. Females and males are equally likely to be affected, and the conditions can skip generations when only carriers are present. Next is Tay-Sachs disease. Tay-Sachs occurs due to mutation in the EXA gene, where the body is unable to produce an enzyme that breaks down fatty substances, resulting in nerve cell damage. This disorder inherited in an autosomal recessive pattern, causing progressive neurological deterioration and is often fatal in early childhood. high such disease is inherited in a autosomal recessive pattern, meaning both parents must carry one faulty gene. If both parents are carriers, their child has a 25% chance of developing the disease, inherits two faulty genes, a 50% chance of being a carrier without symptoms, a 25% chance of being completely unaffected. It affects both genders equally and often skip generations, only appearing if both parents are carriers. Complex disorders. So what is a complex disorders? Complex disorders are medical conditions caused by combination of genetic, environmental, and lifestyle factors, making them challenging to diagnose and manage. Due to their integrate nature, they can present differently in each person, often requiring personalized treatment approaches. So these are the example of complex disorders. Heart disease. Heart disease refers to a group of conditions that impair the heart's ability to function properly, such as coronary artery disease and heart failure. It is influenced by genetic predispositions, lifestyle choices like diet and exercise, and environmental factors such as smoking and pollution. Diabetes. Diabetes is a metabolic disorder where the body has difficulty regulating blood sugar due to issues with insulin production or effectiveness. Its development is influenced by genetic factors, along with lifestyle habits such as poor diet, lack of exercise, and obesity. Asthma. Asthma is a chronic lung condition that causes inflammation and narrowing of the airways, making breathing difficult, and is influenced by genetic susceptibility and environmental triggers, including allergens, air pollution, and respiratory infections.
Well, good evening everyone. Here in the role of genetic mutation in evolution. For mutations introduce new variation within a population. Those variations that provide an advantage for survival and reproduction in a particular environment are more likely to be passed on to the next generation. This process is called natural selection. Mutations are random changes in DNA sequence. They can be beneficial, harmful, or neutral. Beneficial mutation increases an organism's chances of survival and reproduction in a particular environment, while harmful mutation decreases an organism's chances of survival and reproduction. And lastly, neutral mutations have no effects on an organism's fitness. Natural selection acts on these variations favoring those beneficial in given environment. This means that individuals with beneficial mutations are more likely to survive and reproduce, passing on their genes to the next generation. We have Darwin finches. So, Darwin finches, famously observed by Charles Darwin during his voyage on the HMS Beagle. This provides a classic example of adaptive predation. This refers to the rapid diversification of a single ancestral species into multiple descendant species, each adapted to a different ecological niche. On the Galapagos Island, Darwin observed 14 distinct species of finches, all descended from a common ancestor. Each species had evolved unique beak shapes and sizes, perfectly suited to the specific food source available on their respective islands. For example, finches with strong, thick beaks were adapted for cracking seeds, while those with long, slender beaks were better at probing for insects. This diversification of beaks morphology allowed the finches to exploit different food resources, minimizing competition and maximizing survival. To summarize that, Darwin finches, 8 different beak shapes in finches on the Galapagos Island, evolved to exploit different food sources, showcasing adaptive predation. So the next part is peppered moths. The peppered moth, Biston betularia, provides a striking example of natural selection in action. Before the Industrial Revolution, the peppered moth population was predominantly light-colored. blending in with the lichen-covered tree bark. Where they rested, this camouflage is protected from the predators, such as birds. However, as industrial pollution increased, the lichen died off, and the tree bark became darkened by salt. These environmental changes favored the dark-colored moths, which were now better camouflaged against the blackened bark. The lighter-colored moths, now more visible, were more likely to be preyed upon. Over time, the frequency of the darker-colored moths increased in the populated areas, while the lighter-colored moths become less common. This shift in population proportions demonstrates how natural selection can favor traits that enhance survival in changing environments. So, the color of peppered moths shifted from light to dark due to the industrial population, demonstrating the natural selection favoring traits that enhancing survival in changing environment. Next is antibiotic resistance in bacteria. Antibiotic resistance in bacteria is a modern example of rapid evolution. Bacteria reproduce quickly, allowing for the rapid spread of beneficial mutation. When bacteria are exposed to antibiotics, those with mutation that confer resistance to the antibiotics survive and reproduce, while susceptible bacteria are killed off. So bacteria is rapidly develop resistance to antibiotic through mutation, highlighting the speeds of evolution of its implication for humans' health. So the impact of the genetic mutation on the evolution of life on Earth. It all starts with tiny changes in our genes called mutation. These mutations can be like little tweaks to our blueprint, sometimes making us better suited for our environment, sometimes not. So, using examples like Darwin finches, peppered moths, and antibiotic-resistant bacteria to show how these tiny changes can have a huge impact on the survival and success of different species.
Hello, and welcome to this presentation on detection and analysis of mutations. In this discussion, we will explore the key techniques used to detect genetic mutations, including DNA sequencing, polymerase chain reaction, PCR, and gel electrophoresis. These methods are essential in understanding how mutations affect the function of genes and their significant impact in various fields like medical diagnostics, research, and forensic science. Mutations, which are changes in the DNA sequence, can lead to a wide array of genetic disorders or variations, and their detection is crucial for advancing our understanding of genetics. DNA sequencing. DNA sequencing is one of the most powerful tools used in mutation detection. This technique allows scientists to determine the exact order of nucleotides, adenine, thymine, cytosine, and guanine within a DNA strand, revealing any mutations present. The two main forms of DNA sequencing are Sanger sequencing and next-generation sequencing, NGS. Sanger sequencing, known for its accuracy, has been widely used for many years, but it is relatively slow and better suited for smaller-scale projects. On the other hand, next-generation sequencing is faster, more cost-effective, and capable of analyzing large portions of the genome, making it ideal for high-throughput sequencing. This technology is crucial in detecting mutations such as single nucleotide polymorphisms, SNPs, insertions, and deletions, which can be linked to genetic disorders like cancer. In clinical applications, NGS is used to screen for mutations in genes like BRCA1 and BRCA2, which are associated with an increased risk of breast and ovarian cancers. Polymerase chain reaction, PCR. Polymerase chain reaction, PCR, is another essential technique for mutation detection. PCR amplifies specific DNA regions, allowing scientists to focus on particular sections of the genome. It is particularly useful when working with small or degraded DNA samples. PCR uses primers, short sequences of nucleotides that target specific regions of the DNA, followed by repeated cycles of heating and cooling to replicate the DNA. This technique makes it easier to detect mutations even in small quantities of DNA. For example, allele-specific PCR can be used to identify mutations associated with certain genetic diseases, such as the mutation responsible for sickle cell anemia. PCR plays a critical role in both genetic research and clinical diagnostics, providing a reliable method for analyzing specific mutations in DNA samples. Gel electrophoresis. After PCR amplification, gel electrophoresis is often employed to visualize and analyze DNA fragments. This technique separates DNA based on fragment size by applying an electric current to a gel matrix. The DNA fragments, which are negatively charged, move toward the positive end of the gel, with smaller fragments moving faster than larger ones. This separation allows researchers to compare fragment sizes and detect structural changes in the DNA, which may indicate the presence of mutations. For instance, single-strand conformation polymorphism, SSCP, a variation of gel electrophoresis, can be used to detect subtle differences in DNA structure caused by mutations. Gel electrophoresis is a valuable tool for confirming mutation results obtained from PCR or DNA sequencing, and it is widely used in forensic science to compare DNA samples in criminal investigations. Applications in genetic testing, research, and forensics. The techniques discussed, DNA sequencing, PCR, and gel electrophoresis, have a wide range of applications. In genetic testing, they are used to identify mutations that increase the risk of hereditary diseases, such as mutations in the BRCA genes that predispose individuals to breast cancer. This information is crucial for early diagnosis, personalized treatment plans, and preventive care. In research, these techniques allow scientists to study gene function, explore genetic variations, and develop new therapies. By understanding how mutations influence disease progression, researchers can discover novel drug targets and therapeutic approaches. In forensic science, PCR and gel electrophoresis are indispensable tools for DNA fingerprinting and DNA evidence from crime scenes, helping to identify individuals or establish biological relationships.
Moving on, we will now discuss the ethical considerations for genetic mutations. First, we have the CRI-SPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. It refers to a natural system used by bacteria to defend against viruses, which scientists have adapted for gene editing. CRI-SPR is a groundbreaking gene editing technology that allows precise modifications to DNA. It uses a guide RNA to target specific genetic sequences, enabling scientists to add, delete, or alter genes effectively. This tool holds immense potential for applications in medicine, agriculture, and research, but it also raises ethical concerns about genetic modification, including risks of unintended consequences and the moral implications of altering human embryos. The ongoing dialogue around CRI-SPR balances its ultimate benefits against possible societal impacts. Next, we have genetic privacy and consent. Individuals may have their genetic information tested for mutations, raising concerns about who has access to this data and how is it used. Informed consent is crucial to ensure individuals understand the implications of testing and potential consequences. Next is discrimination and stigmatization. There is a risk that individuals with certain genetic mutations may face discrimination in areas like employment, insurance, and healthcare. Protecting against genetic discrimination is essential to ensure fair treatment. Next, we have gene editing and enhancement. Technologies like CRI-SPR raise questions about the ethics of editing genes in humans, particularly regarding designer babies. So, designer babies refers to the concept of using genetic engineering technologies like CRI-SPR to select or enhance specific traits in embryos, such as physical appearance, intelligence, or resistance to diseases. There are concerns about creating socioeconomic divides based on access to genetic enhancements and the potential societal pressure to conform to certain genetic traits. Moving on, we have the impact on future generations. Germline editing, or altering genes in embryos, affects not just individuals but also their descendants, raising questions about the long-term consequences and responsibilities toward future generations. Next, we have biodiversity and ecosystem health. Gene editing in organisms could affect ecological balance and biodiversity, leading to unintended consequences that disrupt natural ecosystems. Lastly, we have moral and religious considerations. Different cultures and belief systems have varying views on the morality of manipulating genetic materials, which can influence public perception and policy.
In conclusion, understanding genetic mutations is crucial for advancing biology, treating genetic diseases, and uncovering evolutionary history. Ongoing research continues to uncover the implications of mutations in health, agriculture, and the future in humanity.